And hello, everyone, and thank you for joining us today. And for today's presentation, it divided into 4 sections: an overview of in vivo CAR-T and LVV manufacturing challenges, quality control assets, and the regulatory requirements for LVV as a drug product. First, I'll provide a brief comparison of ex vivo and in vivo CAR-T therapy. The traditional ex vivo CAR-T therapy involves collecting T cells from patient and modifying them outside the body and expanding them, and then reinfusing the engineered T cells. In vivo CAR-T streamlines this entire process. A viral vector is injected directly into the patient and modifies the T cells inside the body. No apheresis, no cell culture, just a significant simplified workflow. And to clearly illustrate the strengths and the limitations of each approach, I'll walk you through a head-to-head comparison. Logistically, ex vivo therapy requires apheresis followed by 9 to 21 days of manufacturing, whereas in vivo therapy can be produced in under 24 hours and is fully off the shelf. And it also simplifies the cold chain logistics, requiring dry ice instead of liquid nitrogen. And clinically, the in vivo CAR-T has demonstrated a lower CRS incidence to date and may eliminate the need for lymphodepletion. However, the off-target risks must be well managed. And from the commercial perspective, the in vivo CAR-T enables lighter shipping and broader hospital access, expansion beyond Tier 1 cities, and the application in solid tumor and autoimmune diseases. In vivo CAR-T can be delivered via lentiviral vector, AAV, and TLNP and polymers, each with trade-offs in safety, targeting, and durability. Except for lentiviral vector, both AAV and mRNA-based TLNP are used for transient expression because the therapeutic gene cannot integrate into the host cell genome. And then let me focus on the lentiviral vector options. Lentivirus package a single-stranded RNA genome, and that is reverse transcribed and integrates randomly into the host genome. And the particle displays a surface-anchored recombinant binder, typically an anti-CD3 or anti-CD8 scFv. After the binder docks onto the T cell epitope, the targeted VSVG mutant mediates membrane fusion. Once integrated, the transgene is replicated and inherited by every daughter T cell, provide decades of expression. And by abandoning the requirement for VSVG targeting, lentiviral vector can transduce resting T cells. In short, the lentiviral vector platform enables permanent and high-level CAR expression in specific T cell subsets. And on the downside, the random integration may pose safety risks that require long-term monitoring. And here is a snapshot of the LVV-based pipeline. Companies like Interis, Umoja, and Isobiotech are currently in phase 1 trials. They target including CD19, CD20, BCMA, and other even solid tumor antigens. Most use CD3 CD3 or CD8 targeted envelopes to direct the vector to T cells. And here is a similar snapshot for the tRNP-based pipelines. And next, I'll discuss the key challenges in LVV process development. The lentiviral vector's role is to deliver a therapeutic gene into T cells, converting them into tumor-killing agents. Viral vector therefore focuses on two aspects: precise T-cell targeting and potent, durable cytotoxicity. Envelope engineering achieves pinpoint to key T-cell targeting, while the payload design maximizes the potency and the persistence of the engineered tumor-killing response. On the lentiviral vector surface, the binder, usually anti-CD3 or anti-CD8 SCFV, acts as a GPS that identifies its target antigen in the bloodstream. The fusogen is an engineered VSVG mutant whose receptor binding domain has been removed, rendering it unable to bind its receptor LDLR and active only after SCFV docking brings the viral and cell membrane into proximity. And for the payload, both promoter and the signal peptide critically govern the CAR expression levels and by extension directly influence the lentiviral vector titers and the overall yield. Promoter selection depends on whether the CAR expression always on T cell only or antigen gated expression is desired. The signal peptide determines whether the protein efficiently enters the secretory expression pathway, directly affecting CAR surface density and exhaustion kinetics. And the transmembrane domain anchors the CAR in the membrane and governs whether it partitions into lipid rafts or is cleaved by gamma secretase, thereby shaping signal strength and duration. And the Umoja and the isobiotech illustrate two distinct optimization paths. Umoja focuses on the payload engineering, while isobiotech re-engineers the particle surface. Umoja's UBVV100 uses an anti-CD3 for homing and CocoG for targeting, for entry. And in addition to VSVG mutant, envelopes such as CocoG and those derived from Nipah virus and measles virus are also used for in vivo application, as VSVG is sensitive to human serum and exhibits low transduction efficiency in resting T cells. Beyond the conventional CAR cassette, its transfer plasmid encodes a rapamycin responsive cassette that permits ex vivo and in vivo expansion of engineered T cells without additional cytokines. And isobiotech, in contrast, retain a classic BCMA CAR payload, but re-engineers the particle surface, the binder, and plus the target VSVG mutant are combined with MHC1 knockout on the producer cells and the CD47 overexpression on the viral vector membrane. These two modifications that blunt anti-vector immunity and reduce macrophage phagocytosis and prolong the circulation half-life of the lentiviral vector in vivo. The challenges of lentiviral vector manufacturing is to meet demands for product safety, yield, and high purity through vector design and process optimization. Compared to the standard 4-plasmid transient transfection system, in vivo LVV can continue to use GAG-POL plasmids and GAG-POL plasmid and RAB plasmid. If a non-integrating or site-specific integration design is desired, the POL sequence must be modified, and the VSVG plasmid can be replaced with plasmids expressed bander and VSVG mutant for in vivo CAR T application. And the envelope plasmid can further be optimized to enhance T cell tropism or enable transduction to resting T cells. For further enhanced LVV safety, strategies like site-specific integration and non-integrating LVV molecular switches and insulators require modification of both hyper and transfer plasmids. In the transient transfection system, envelope density is critical for transduction efficiency, and because the envelope plasmid design and its regulatory elements govern gene expression, and because each customer project may employ different binders and envelopes, plasmid ratio and total plasmid amount become key process parameters that must be optimized. Consequently, plasmid ratio screening is required before any packaging test. And for the host cell protein, it's an important quality attribute, and a higher purity is always desired. However, because the lentiviral vector acquires its envelope from the host cell membrane, and a fraction of these HCP remained product associated and co-enriches as lentivirus concentration increases, and therefore a scientific justified specification for HCP must be established. And the envelope sequence can impact the lentivirus aggregation and should be well optimized, as process parameters cannot effectively prevent aggregation caused by envelope-mediated binding interaction. And as a drug product, safety and safety specification must be met, and the formulation buffer should carefully optimized for organ and tissue-specific injection. And the expression of CAR during lentivirus production can cause off-target binding. For example, the CD19 CAR displayed on the vector surface the CD19 CAR displayed on the lentiviral vector surface can bind B cells, creating non-specific CAR B cells. For ex vivo CAR T process, non-specific B cells can be controlled by process optimization, whereas for in vivo application of target binding to B cells may be a concern for safety and efficacy. As noted, the current well-validated CAR T payload are CD19 and CD20. Therefore, repressing GI expression is critical for these projects. And for the in vivo LVV process optimization, two aspects require particular attention. Receptor-targeted LVV are highly sensitive to SCFV design. Envelope density must be precisely tuned to prevent steric hindrance, and we therefore optimize plasmid ratio to achieve ideal surface density in transient transfection system. And a stable producer cell line can also be employed to increase process robustness. And the rational design of SCFV sequence eliminates the stretches that drive non-specific crosslink, thereby avoiding syncytium formation in the packaging cells, which is a major cause of reduced LVV yield. And such crosslink can also trigger the LVV aggregation during production, even in the higher final concentration. So removing these motifs simultaneously boosts both titer and the purity. And next, I'll walk you through our process development workflow based on our experience. And our optimization strategy uses a small scaled-down model. We first maximize crew titer by optimizing transfection conditions using DOE, including plasmid ratios, cell density, and harvest time. And we then screen fed strategies and refine bioreactor parameters. And a final confirmation run demonstrate a scalable performance once all conditions are fixed. And for in vivo LVV application, multiple envelope and binder choices create significant downstream challenges. And therefore, downstream process road screening is beneficial for determining an optimal workflow. And during optimization, we focus on filtration for debris, cell debris removal, and UFDF for concentration and buffer exchange, and chromatography for impurity removal. And all steps are optimized in scaled-down models before scaling up. And after the overall downstream process optimization, the recovery and impurity levels could meet our platform standard. And for the in vivo LVV application, our ProBio provides LVV production service via suspension system with a crew titer of 1E7 to 1E8 TU per mil at a scale up to 200 liter. And the workflow for in vivo LVV application is outlined below. And the production uses our proprietary cell line, PowerS293T cell line. And as noted, multiple plasmid transfection for in vivo CAR-T application can compromise transfection efficiency and make transfection parameters optimization critical. And based on our experience, the maximum plasmid amount we have been conducted is 6. And there was no significant difference with the 4 plasmid transfection system, and the harvest titer is comparable. And since the different envelope can affect LVV stability, and the formulation buffer must also be optimized. And next, I'll cover the quality control assay for in vivo LVV application. And the release criteria are displayed in the table, covering physical titer, purity, and safety testing. In vivo LVV drug products require additional quality control measures compared to drug substance, including tests for visible particulates, fuel volume, particulate matter, and the particle size. And we developed comprehensive mechanism of action, biological activity, assay that span vector integration and transiting expressions of final therapeutic activity. And our strength is proven execution. Over 100 biological activity methods have been successfully developed and delivered. And another key strength is our fully validated platform, which enables a platform plus PSQ model that delivers faster IND submission at lower cost. And finally, regulatory requirements are a critical consideration. For in vivo LVV application, lentiviral serves as a drug product to meet gene therapy regulatory requirements. Specifications for host cell DNA and the fragment size must be met, and the endotoxin limits should be established based on the product type. And the insoluble particle specification must comply with USP 789 and USP 788 based on product type, tested by light obfuscation and microscopy methods. Since lentiviral vectors are fragile, requiring continuous downstream processing, and this approach is acceptable by both FDA and EMA, and obviously need for drug substance freezing and stability studies. And that concludes my presentation, and I would be happy to answer any questions.
Thanks for Dr. Xuan's excellent presentation. We will now open the floor for questions. Please type your questions in the chat panel. Additionally, we warmly invite you to follow our LinkedIn account profile, and the Korean attendees can also scan the QR code on the screen to follow our Kakao channel, where we will regularly share online resources and updates of our recent events. Okay, let's take a look at the first question. How do you quantitatively assess envelope density on the LVV surface during process development? And what is your target range for optimal performance? Hi, Dr. Xuan, could you help on this question?
Okay, thank you for this question. And for the envelope density on the LVV surface can be quantified by nanoflow cytometry, and the target range is project specific. So the plasmid ratios must be optimized during the process development stage.
And the second question is, are stable cell lines beneficial for in vivo CAR-T LVV production?
Yes, because the envelope density is critical for LVV transduction, and a stable cell line provide better batch-to-batch consistency than transient transfection system. And the stable integration ensures uniform envelope expression across production batches. For example, isobiotech integrates the binder directly into the HEC293 genome.
Thanks for your answer. And next question is, do you have any IP-free packaging plasmid for in vivo CAR, like fusogen, VSVG, MUT, and binder?
No, right now we don't have any IP-free packaging plasmid, and the envelope plasmid should be provided by client for testing.
Thank you. And the next question is, this presentation slides will be shared later. I can take this. we have recorded this webinar, so the recording link will be shared with everyone about 2 days later. Yeah, so you can find the recording for further study. And the following question is, how do you think the possibility of transgene integration of in vivo CAR T when using lentiviral system? Hi, Dr. Xuan, could you help on this question?
Yes. Because for using lentiviral vector system, we hope the transgene integrated into host cell genome to get prolong the therapeutic gene expression.
And the next question, for the CAR T products, where the manufacturing process and even the formulation can be changed, how do you approach analytical method validation? Are traditional validation still applicable, or is a more flexible life cycle-based strategy expected?
For the in vivo LVV, we may use a project-specific formulation buffer. So I think for for this project, the QC method should be validation project-specific.
Thank you. Next question is, what are the titer difference between a 4 plasmid and the 5th plasmid system?
I think for the 5th plasmid system, there was no significant difference for the titer, but it should be optimized the transfection system to get a better performance. And if the titer increased to 8 or 10, I think the transfection efficiency should be decreased significantly.
Okay. And do you think about LNP or nanocarrier instead of LVV for in vivo CAR T cells?
Yes, it can use for in vivo CAR T, but for the lentiviral vector, the CAR expression set can be integrated into host genome, and during the T cell expansion, the engineered T cell can the duration of the CAR expression is longer than the mRNA, the tRNP-based therapy.
We can also find more details in the following talk presented by Dr. Zhang Xu later. Yeah, so maybe we can hear any more thoughts from Dr. Xu later. And the next question is, could you comment more on using mRNA-LNP as a delivery system? Is there any plan to explore this modality more in the future?
I think Dr.
Yeah, maybe
Sorry.
Dr. Xu can take this question later.
Yes.
Okay, yeah, so that's all the questions I see by now. If you have further questions, you can email us and we can reply you later. And thanks for Dr. Xuan.